Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Illumina Sequencing. Comprehensive guide to principles, process, advantages, and applications. Table of contents. Introduction. Principle. Steps. Process. Advantages. Limitations. Applications. Introduction. Illumina Sequencing is a leading next-generation sequencing (NGS) technology that employs sequencing by synthesis to identify individual DNA bases as they are incorporated into a growing DNA strand. This technology uses advanced instruments capable of analyzing millions of DNA sequences simultaneously. Initially developed by Solexa and acquired by Illumina in 2007, Illumina Sequencing has become popular for its high-throughput capabilities, which make it faster, more accurate, and more cost-effective than traditional methods like Sanger sequencing. Although Illumina sequencing shares some principles with Sanger sequencing, such as using fluorescently labeled nucleotides to detect individual bases, it differs in scale. Unlike Sanger sequencing, which sequences one DNA fragment at a time, Illumina technology is massively parallel, allowing millions of DNA fragments to be sequenced simultaneously in a single run. Illumina offers a range of platforms tailored to different research needs, from benchtop to large-scale applications. Compact benchtop sequencers, like the MeSeq and MiniSeq systems, are ideal for laboratories with smaller sequencing demands. In contrast, high-capacity sequencers, such as the NextSeq and NovaSeq systems, are designed for large-scale sequencing projects. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Principle of Illumina Sequencing. Illumina sequencing is based on the principle of sequencing by synthesis, SBS, where DNA bases are identified as they are incorporated into a growing DNA strand. This process uses fluorescently labeled reversible terminator nucleotides. Each of the 4 DNA bases has a distinct fluorescent dye, allowing the system to capture images of the fluorescence emitted from each added nucleotide and determine the sequence of the DNA fragment. A key component of Illumina sequencing is bridge amplification. In this step, DNA molecules with ligated adapters serve as templates for repeated amplification on a solid surface, such as a glass slide. This slide is coated with oligonucleotides complementary to the adapters, enabling DNA to form clusters from each fragment. The clusters intensify the signal, making it easier to distinguish between DNA bases by color. During sequencing, nucleotides with unique fluorescent tags are added one at a time, incorporated, and detected in real time. These nucleotides also act as temporary terminators, allowing only one base to be added per cycle, which enhances accuracy and minimizes errors. After imaging, the terminator is cleaved, and the next base is incorporated. This cycle continues until the entire DNA fragment has been sequenced. Illumina sequencing steps. Steps. Process of Illumina sequencing. First, nucleic acid extraction. The first step in Illumina sequencing involves isolating genetic material from the sample. This extraction process is crucial as the quality of the nucleic acids directly impacts the sequencing results. After extraction, a quality check is performed to ensure the purity and accurate quantification of the nucleic acids. Purity is typically assessed by UV spectrophotometry, while nucleic acid concentration is measured using fluorometric methods. Second, library preparation. Once the nucleic acids are isolated, they undergo library preparation, creating a set of adapter-ligated DNA fragments ready for sequencing. This process begins with DNA fragmentation, breaking the sample into smaller fragments through methods like mechanical shearing, enzymatic digestion, or transposon-based fragmentation. The fragments are then prepared for adapter attachment through end repair and A-tailing. Adapters containing sequences for binding to the sequencing flow cell are added to both ends of each fragment. These adapters may also include barcodes, enabling simultaneous sequencing of multiple samples and differentiating them during analysis. Third cluster generation by bridge amplification. The prepared DNA library is loaded onto a flow cell where amplification and sequencing occur in small lanes. DNA fragments bind to complementary primers attached to the solid surface of the flow cell and undergo bridge amplification. During this step, each DNA strand forms a bridge structure on the chip with the help of forward and reverse primers. Each bridge is then amplified, creating dense clusters of DNA at each spot. Cluster generation completes when there are sufficient copies of DNA at each location to produce a clear signal for sequencing. Fourth, sequencing by synthesis, SBS. With clusters in place, sequencing by synthesis, SBS, begins. Fluorescently labeled nucleotides are added one by one to the DNA strand. Each incorporated nucleotide emits a specific fluorescence, which allows the system to identify it. The sequence of each DNA fragment is determined through multiple cycles of this process. Fifth, data analysis. After sequencing, the collected data are processed and analyzed using bioinformatics tools. Fluorescent signals from each cycle are converted into base sequences. Bioinformatics tools then clean and organize this data, preparing it for further analysis. Sequences can be aligned to a reference genome or assembled if a reference is unavailable. This process helps in identifying variants, mapping genes, and supporting further analyses. Some Illumina instruments include user-friendly analysis software, allowing researchers without specialized bioinformatics expertise to interpret sequencing data for insights like pathway analysis, biomarker identification, or gene function prediction. Advantages of Illumina sequencing. Enables parallel sequencing of millions of DNA fragments, generating extensive data in a single run, ideal for large-scale projects like whole-genome or transcriptome sequencing. High-throughput approach that saves time and resources, delivers highly accurate results with minimal sequencing errors, ensuring reliable data. Supports rapid sequencing, making it particularly valuable in clinical applications. Cost-effective for large-scale projects, providing an affordable alternative to traditional methods like Sanger sequencing. Compatible with both single-read and paired-end libraries. Flexible platform supporting a variety of sample types and library preparation methods, suitable for analyzing whole genomes or targeted regions. Adaptable to different applications and research needs. Limitations of Illumina sequencing. Generates short reads, making it challenging to assemble complex genomes or highly repetitive regions. While cost per base is low, the initial investment in sequencing equipment and maintenance can be costly for smaller labs. Produces a large volume of data that requires powerful computational tools and specialized expertise for analysis. Faces challenges with loss of synchrony during sequencing, as not all molecules in a cluster incorporate nucleotides simultaneously, potentially affecting accuracy. Risk of overclustering if too much DNA is loaded onto the flow cell, which can reduce sequencing quality and accuracy. Applications of Illumina sequencing. Supports a wide range of applications, including genomic sequencing, RNA sequencing, metagenomics, and ChIP-seq. Ideal for large-scale genome projects, targeted resequencing, and epigenetic studies, enabling exploration of genetic modifications and their roles in diseases. Useful in microbiome research. amplicon sequencing can analyze microbial communities and microbial genomes, aiding in outbreak tracking and understanding antibiotic resistance. Enables cancer research by identifying mutations, tracking disease progression, and uncovering potential therapeutic targets. Valuable in environmental studies for assessing biodiversity through sequencing of DNA from mixed environmental samples. Applied in forensic science to analyze genetic material from crime scenes, assisting in suspect identification and determining relationships between individuals. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!